Hi, I'm Simon Harrison. I'm the Director of the Centre of Excellence for Cellular Immunotherapy at the Peter MacCallum Cancer Centre in Melbourne. I'm one of the leading myeloma specialists here. MRD is really an important response criteria, as the treatments that we use in myeloma are much more powerful, and therefore we need better discriminators to measure the success of the therapy, but also they're a predictor of progression-free and overall survival, and therefore they are helping us have early readouts of the studies. And there are a number of current or recently reporting trials that are using MRD really to help guide treatment strategies. The first of which was the MASTER study, and that's looking at changing the treatment based on achieving MRD response and being able to stop treatment after stem cell transplant or after a number of rounds of consolidation and maintenance treatment, to really see whether the achievement of MRD is an endpoint on its own and can guide when you can stop therapy and give patients a break from treatment. I guess, in my opinion, MRD is really the starting point to try and achieve long-term functional disease control, and it'll be interesting to see whether these strategies bear fruit. There are a number of other trials looking at combination treatment, such as the GMMG concept study using a combination of isatuximab couples with lenalidomide and dexamethasone, where they're looking to improve the response rate in high-risk patients with high-risk cytogenetic abnormalities and high-risk disease, to again really see if you can drive long-term disease control by achieving MRD negative responses. And this is an interesting approach. And then other studies like the Griffin study using daratumumab in combination with VRd is looking to see whether achievement of high-quality responses can be recapitulated in it using a different upfront treatment, and trying to see whether these treatment strategies will allow patients to achieve functional cures. The final piece of data that's come up recently is an analysis of the IFM 2009 trial, which showed that even in patients who achieve MRD negative responses, whether they had a transplant or not, the transplant patients did better. And therefore, it would seem that there is even better quality of responses, even when you're looking at MRD at 10 to the minus 6. And this probably points to one of the weak points of MRD measurement as we currently use it, because it's really only measuring one compartment, such as the bone marrow. And there are maybe other reservoirs of relapsed disease in extramedullary compartments that are not being measured accurately. And we need to think about how we integrate other high quality measures of response, such as PET, ctDNA, and mass spec into the treatment algorithms. So in myeloma, we're fortunate to have a number of immune-based therapies at our disposal. And these are the drug antibody conjugates, naked antibodies, bispecific antibodies, and CAR T cells, which are really one of the new and exciting therapies available. There's naked antibodies in the form of daratumumab and isatuximab have been around for a while and seem to add around 20% improvement in response rate. And this is kind of fairly similar to the impact of rituximab in other diseases like lymphoma. Moving on from there, the drug antibody conjugates are really quite interesting in belantamab delivers a payload into the bone marrow to try and improve the cell kill associated with antibody therapy. And the interesting thing is, as a single agent, the response rate is relatively modest, but if you do respond to these treatments, it seems to have an increased immune control, and there's a long tail on the curve of these studies. And there are a lot of studies looking at combination approaches in the DREAM series of trials to try and improve these response rates. Bispecific antibodies really are a halfway house between antibody therapy and CAR-T, where one end targets an antigen on the myeloma surface, and there are a number, including BCMA, that are being investigated at the moment. And then the other end binds to CD3 on the T cells to activate the T cells when the antibody binds to the myeloma and allow the T cells to kill by degranulation, expand, activate, and go on and perform serial killing, hopefully. The downside of this is once the antibody is the half-life of the antibody is achieved, the amount of antibody disappears in the circulation, and therefore patients need to be redosed. The good thing about that is that there's not an exponential increase in the cytokine release and neurotoxicity signals associated with these. So these are relatively short-lived and can be easily treated with steroids. CAR-T cells are really the ultimate in immune-based therapy at the moment. And there are a number of these agents that are now in routine clinical practice with an FDA-approved agent available in the US targeting BCMA. And these cells are manufactured in a laboratory, and a receptor mainly targeting BCMA at the moment is inserted with a co-stimulatory domain. And again, when the cells bind to the target antigen, this triggers T cell activation, expansion, and essentially engraftment to try and produce long-term memory once the bulk of the tumor has been extinguished. And I think this is really an important development in the treatment of myeloma, with very high response rates being seen across the board in these trials. And in the relapse refractory setting, very long-lasting responses compared to standard therapies available up until this point. So I think immune therapies are here to stay in myeloma. There's a number of different modalities by which they can be delivered, and they're really achieving high levels of response in the relapse refractory population that are really unprecedented up until this time point. And there's a move to bring these agents into earlier lines of therapy and up front, and really challenge the treatment paradigm, such as trying to see if we still need transplantation in the treatment of myeloma.